Good afternoon to all the students of BPC first standard. So today we are going to look into histology and anatomy long answer type of questions. So every year in your IPE paper there is a definite question on anatomy, histology and anatomy of flowering plants, where either you can get the comparative analysis of dicot monocot root, dicot monocot stem or dicot monocot leaf, or else you can get a question to elaborate or to brief the description about the anatomy of dicot stem or monocot stem, dicot root, monocot root or dicot leaf or monocot leaf. So here if you see describe the internal structure of dicot root. About dicot root we have to describe the internal structure of it. So the internal tissue organization of dicot root. Here first if you see in the diagram of dicot root, that is sunflower, anyhow the dicot root we have exercised in the previous class. Today we will start with monocot root, okay. Yeah. So the next question is about describe the internal structure of monocot root. To describe the internal structure of monocot root, the answer description will be same as that of dicot root with few changes, that's it. Epidermal description will be same, cortex is parenchymatous, endodermis have Casparian stripes. Next to that you will have pericycle. In the stellar region which includes pericycle, vascular bundles and pith. Vascular bundles, here you can see polyarch condition and a broad pith you can observe, and the absence of cambium. These are the only three changes that you observe in difference with dicot root, else everything is same, rest everything is same. Here you see into the description the transverse section of a monocot root is differentiated of course into three zones, same as that of dicot root, okay. That is epidermis, cortex and stele. First you see epidermis, this is the outermost layer of the root, uniciliate layer, this is uniciliate or single layered or uniciliate, okay, uniciliate or single layered, and it is composed of elongated compactly arranged cells. Many epidermal cells will protrude into unicellular hair-like projections. That is the reason epidermis has, root epidermis is also addressed as epiblemma, piliferous layer or rhizodermis. Clear, right? We have learnt in the previous answer also. Epidermis is also addressed as rhizodermis, piliferous layer or epiblemma. Fine, right. So many epidermal cells will protrude to unicellular hair-like projections, those are root hairs. Epidermal cells and root hairs, they help in both water absorption and mineral absorption from soil. Cuticle and stomata are completely absent here, okay. Cuticle and stomata are absent in the epidermis. This provides protection to the inner tissues, okay. This will provide, epidermis layer will provide protection to the inner tissues. Next you see cortex region. These are composed of several layers of thin walled parenchymatous cells with least intercellular spaces. The innermost layer of the cortex is endodermis. Endodermis here, single barrel shaped structure, okay, endodermis is single barrel shaped structure. This does not have any intercellular spaces. So the radial and tangential walls of endodermis is made up of what depositions? Beta? Suberin depositions. These suberin depositions are impervious for water absorption, okay. The cell wall is impervious for water conduction, okay. Next you see stele region is differentiated into pericycle, vascular bundles and pith. Pericycle is the outermost layer, okay, next to the endodermis. So the innermost cortical layer here is what endodermis, and outermost layer of the stele is what? Outermost layer of the stele is pericycle, okay. Next you see this helps in formation of lateral roots, and in monocot root if you observe, pericycle does not take part in the secondary growth. So from pericycle only lateral roots will arise. Lateral roots arises. no cambium. Fine, right ma? Next, vascular bundles. If you see vascular bundles, they are radially arranged of course, and xylem and phloem are present separately as at different radii, that is why we call it as radial type of vascular bundle arrangement. And in dicots you can see diarch to tetrarch condition, which are 2 to 4 vascular bundles, but here in monocots you can see more than 6 vascular bundles, which is polyarch condition, more than 6 vascular bundles. Cambium is absent between xylem and phloem. Vascular bundles are of closed type. In dicot root, cambium is present between xylem and phloem, that is open type of vascular bundle. Here exarch xylem is wherein protoxylem is towards the periphery and metaxylem is towards the center, which is the exarch type of vascular bundles. This is same dicot root and in monocot root. Next, conjunctive tissue is present between xylem and phloem. Okay, conjunctive tissue is present in between xylem and phloem, and in dicot root from that conjunctive tissue only beta vascular cambium will arise. Here secondary growth is usually absent in monocot roots. Okay, secondary growth is absent in monocot roots. Next is what? Medulla or pith. Medulla or pith. Central part of the stele is occupied by medulla or pith. Pith is large and well developed in the monocot root. It is small and inconspicuous in dicot root. So, the best mnemonic to remember the sequence, to present the answer in a sequential manner. Every clever engineer plans very massive project where E stands for epidermis over here. So, here you see see carefully for monocot root to describe it in a clear way. For monocot root, to describe it in a sequential pattern, you remember this mnemonic, you can easily present the answer. E for what? Epidermis, then comes the cortex region, E endodermis, P pericycle, vascular bundles, medulla or pith. This will be the sequential description. First you will describe about epidermis, then the cortical region which includes general cortex and endodermis, and then stellar region which includes pericycle, vascular bundles, medulla or pith. Clear, right? Yeah. Next you observe, describe TS of dicot stem, transverse section of dicot stem. This question has appeared in many years like June 23, March 2023, October 2021 and May 2018. Okay, these many times the question has appeared. The transverse section of young primary dicot stem is then differentiated into epidermis, cortex and stele. Okay. So, here you see epidermis, cortex and stele. Yeah. First, epidermal hair has multicellular hair-like projections. I will first explain you with the diagram and then we will be going for the answer. First, you see the sequential description about epidermis. Epidermis, unlike the root, here epidermis will have multicellular hair-like projections. Those multicellular hair-like projections are called as trichomes. And then cortex region is divided or differentiated into outer hypodermis, next to hypodermis you have general cortex, next to general cortex you have endodermis. Fine, right? So, you have hypodermis, general cortex and endodermis. Hypodermis in dicot stem is made up of collenchyma. Hypodermis in dicot stem is collenchymatous. It is made up of collenchyma. Next, few of the cells of collenchyma, if they are photosynthetic, if they consist of chloroplast, then you can address it as chlorenchyma. General cortex is parenchymatous over here, and last layer of cortex region is endodermis. Last layer of the cortex region is endodermis over here. Next, in the stelar region, stele of course includes what ma? Stele includes pericycle, vascular bundles and pith. Fine. So, pericycle here, endodermis consists of starch sheath, you remember in dicot stem. Hypodermis is made up of collenchyma. If few of the cells are having chloroplast and conducting photosynthesis, it is also known as chlorenchyma. And next to that, the general cortex is made up of parenchyma. After that, endodermis. Endodermis consists of starch sheath. Fine, right? Endodermis here consists of what ma? Starch sheath. Next to that you have bundle sheath cells. Okay, and stelar region includes what? Pericycle, vascular bundle and pith. So here pericycle is again sclerenchymatous. It has semi-lunar patches, semi-lunar sclerenchymatous patches. So pericycle here it is again sclerenchymatous. Vascular bundles has both xylem and phloem. As it is dicot stem, it has cambial ring in between vascular bundles. That cambial ring is of two types, intrafascicular cambium and interfascicular cambium. Pith or medulla, which is at the center, it is parenchymatous. From pith you can also have what? Medullary rays in the dicot stem. This is the brief description about dicot stem. So, in dicot stem, the regions, each region is composed of different types of tissue. So, there will be more elaborate explanation when you give about dicot stem. When you choose the answer of dicot stem, you have to give more elaborate explanation beta. For that, you first present the diagram. After diagram, you start on describing the answer. So, look at it. Yeah, first it is epidermis, which is the outermost uniseriate layer, okay, single layered, elongated, and here you see closely arranged cells. Cells are closely arranged here. Cells are closely arranged here. This is without intercellular spaces. And above the epidermis, this is covered by a thin cuticle which consists of cutin, which is impervious for gaseous exchange again. Next, young stems, epidermis have few stomata in the young stems and it contains multicellular hair-like projections called as what? Trichomes. Cuticle and trichomes, they will reduce the water evaporation and they will protect the stem from higher temperatures. Stomata in the epidermis, they will allow what? Gaseous exchange and they will regulate the transpiration as well. Stomata in the epidermis, they will allow gaseous exchange and they will regulate what? Transpiration. Next you observe ma cortex. Cortex is located below the epidermis and this lies between the epidermis and pericycle of the stele. That cortex is further differentiated into what? So, this cortex will consist of smaller in width, it is smaller in width when compared to stele. This point if you want to write, you write, if not you can leave it ma, no problem. But this is important, cortex is differentiated into how many zones, that is important. It is differentiated into hypodermis, general cortex and endodermis. Okay, hypodermis, general cortex and endodermis. Here you see hypodermis first. Hypodermis, it is the outermost part of the cortex region. This is composed of few layers of collenchymatous cells. Important, very important. The examiner will be looking for this only. Each layer or region is composed of what type of tissue is very important, of which dicot stem it is furthermore clear description is required. So, here you see provides what? Mechanical or tensile strength to the young stem and this also has assimilatory function, okay, and this conducts what? Photosynthesis. Next, general cortex you see ma, present between hypodermis and endodermis, composed of parenchymatous cells. Composition of each layer is important. Okay, this is composed of thin walled round parenchymatous cells. When parenchymatous cells are there, of course it can have minute intercellular spaces. Outer layer may contain chloroplast and which are photosynthetic. So this is called as what? Chlorenchyma. Fine, right? So the hypodermis is made up of, collenchyma, general cortex parenchymatous, outer region of parenchyma, if they are conducting photosynthesis, we will call it as what? Chlorenchyma. Next, inner layers contain what? Leucoplast. Leucoplast will store the food material. Next is endodermis. Endodermis is the innermost layer of the cortex. Endodermis is the innermost layer of the cortex. Cells are barrel shaped here, they are compactly arranged, and cells contain vacuolated protoplasm. They will contain vacuole inside it. Due to large starch storage, endodermal cells are also called as what? Starch sheath. Okay, they are also called as starch sheath because they contain starch. They store starch in them. So clear with the cortex, right? In cortex region we have, it is differentiated into 3 regions. What is that? Outer is hypodermis, which is made up of collenchyma. Next to that, general cortex, which is made up of parenchyma, of which outer layers of that parenchyma are chlorenchyma. We will call it as chlorenchyma because it contains chlorophyll in it. After which you have endodermis, which stores starch, and that is why you will call it as what? Starch sheath. Next you observe. Next is what? Steel. Central cylinder is concerned with conduction of water and minerals. This occupies large portion of stem's internal structure. So this is differentiated into what? Pericycle, vascular bundles, medulla and medullary rays. First you see pericycle, which is the outermost layer of the steel. This is present as what? Semilunar patches. Semilunar patches or semicircular patches of sclerenchyma, made up of sclerenchyma, that is in between endodermis and vascular bundles. Vascular bundles are arranged in the form of a ring, which is called as U-steel. Bundles are what? Conjoint, collateral and open. Okay. Vascular bundles are conjoint, collateral and open. Next, conjoint, if you see xylem and phloem, they lie together in the same bundle, okay, at the same radius. Xylem and phloem are present at same radius. Fine, right? And collateral, what does it mean by collateral? Xylem and phloem, they are present at the same radius, side by side to each other. They are present at side by side to each other. Open type, presence of cambium in between xylem and phloem. Protoxylem is towards pith, metaxylem is towards the periphery. This is called as what? Endark pattern of xylem development. And here xylem contains of all the 4 types of tissues in angiosperms: tracheates, vessels, xylem parenchyma and xylem fibers. Phloem also consists of 4 types of tissues, that is sieve tubes, companion cells, phloem fibers and phloem parenchyma. Next you see medulla or pith. Medulla or pith, of course it is composed of parenchyma. Remember plant body is abundantly composed of which type of simple permanent tissue? It is composed of what? Parenchymatous tissue. So the medulla or pith is composed of large number of rounded parenchymatous cells with intercellular spaces are present. Okay. This occupies the central part of the steel and is very large. This serves as what? Storage site for food materials. Next you see medullary rays. Okay. Medullary rays, these are found between the vascular bundles as a few layers of radially placed living parenchymatous cells. This will facilitate lateral conduction of water and nutrients. This will help in initiating the secondary growth as well, medullary rays. How you can remember the clear description of all this? Okay, so the mnemonic which I can give you here is: every clever hero gives excellent strong points more marks. Here you see epidermis, and in the cortex region you have hypodermis, general cortex, endodermis. In the stellar region you have pericycle, medulla and medullary rays. Here itself you give on the composition of each tissue as well. Here epidermis is composed of parenchymatous cells, and it is having cuticle as well as trichomes. Fine, right. Next, cortex region, hypodermis is made up of collenchyma, and then general cortex is parenchymatous, and then endodermis consists of starch sheath. Next, in steel if you see, pericycle has semilunar patches, which are sclerenchymatous. Medulla or pith at the center, which is made up of parenchyma. Medullary rays are also made up of parenchyma, and they also take part in the secondary growth. This is the composition of each tissue. You can take it down or take a screenshot of it better. It is also enough. Next. So, the next question is about what? TS of monocot stem. In monocot stem, if you observe, so in the TS of young monocot stem, the tissues, the broad description that you have seen in the dicot stem will not be present in the monocot. Directly from the diagram itself I will be explaining you, because it just has what? Epidermis, hypodermis, ground tissue and vascular bundles, that is it. So, look into the description here carefully, ma. Yeah. So, as how you have seen, epidermis is having cuticle layer above it. Same in monocots you have cuticle, okay, and it also has hair like projections, that is trichomes. And here if you see, the general cortex or the cortex region will have what? Hypodermis and general cortex. Hypodermis here is made up of sclerenchyma. Hypodermis here is made up of what? Sclerenchyma, and general cortex is parenchymatous. It is made up of parenchyma. Fine. Hypodermis, which is made up of sclerenchyma, general cortex, which is made up of parenchyma. And few of the cells, like of above layered parenchymatous cells, they can contain chlorophyll, and you will call it as what? Chlorenchyma. Here in the general cortex, after which you do not have any endodermis, there is no endodermis, no pericycle, no pith and no medullary rays. Fine, right? So, here you see, it is only completely general cortex which is made up of parenchyma, and here you have vascular bundles which are arranged as patches, okay, they are scattered in their arrangement in the general cortex. How these vascular bundles have originated? These vascular bundles are schizolysigenous in origin. Schizolysigenous in origin, which means schizogeny, which means the cells get separated, and in that intracellular spaces the vascular bundles get originated, and lysogeny after cell death, the large spaces which are created, it is because of both the things. One is schizogeny, large intercellular spaces, and lysogeny, because of the death of the cells, large intercellular spaces are created, and in these spaces vascular bundles are originated. That is the reason we will call it as schizolysogenous in origin. And here vascular bundles have both xylem and phloem, which are arranged conjoined and closed way. There is no cambium here, there is no cambium. Vascular bundles are conjoined and closed. And here if you see, metaxylem is towards periphery and protoxylem is towards the center. This type of vascular bundle arrangement is called as what? Endark type of vascular bundle arrangement. So this is the brief description about the diagram. Once you are okay with it, then we will be going for the answer. Okay. Look into the sequence of the answer here. First, epidermis. Epidermis is the outermost single layer of elongated, compactly arranged cells. Okay, they are elongated and compactly arranged cells without any intercellular spaces. Externally, they are covered by what? Cuticle, and this contains stomata surrounded by dumbbell shaped guard cells, because stomata in monocots will have what guard cells ma? Dumbbell shaped. Stomata in dicots will have what shaped guard cells? Bean shaped guard cells, right? And trichomes are absent over here. And functions, if you see, epidermis will protect inner tissues from mechanical injuries and water loss. This will facilitate gaseous exchange and this will regulate the transpiration via stomata. Next you see hypodermis. Cortex is represented by sclerenchymatous hypodermis. This provides what? Mechanical strength to the stem. Next is ground tissue. Ground tissue is composed of large parenchymatous cells. Here intercellular spaces are present. Parenchyma cell size will vary. From central cells are large and peripheral cells will be small. That will be the variation of parenchymatous cells here, because it is only general cortex layer which is present. Okay. And here endodermis is absent. Okay. Next is what? Vascular bundles. How vascular bundles are arranged? Numerous vascular bundles are arranged. They are scattered in the ground tissue completely, and this condition is called as what? Atactostele. Okay, this condition is called as atactostele, where vascular bundles are completely scattered in the ground tissue. And here if you observe the structure, each vascular bundle is surrounded by sclerenchymatous bundle sheath. Okay, each vascular bundle is surrounded by sclerenchymatous bundle sheath, and the type, if you see, bundles are conjoined, closed. Conjoined where xylem and phloem are at the same radius. Closed, cambium is absent. Okay. Next you see, peripheral vascular bundles are arranged in the smaller size and centrally located will be larger ones. Phloem parenchyma is absent. Sieve tube elements and companion cells are present from phloem. Xylem consists of tracheids, vessels, xylem fibers and xylem parenchyma. All the tissues of xylem are present in the monocot stem. Water filled lysogenous cavities are found within the vascular bundles. These cavities are known as what? Protoxylem lacunae. This will develop due to breakdown or lysis of one or more protoxylem elements. That is why how vascular bundles development is there here, schizolysogenous in origin. And then pericycle, medulla and medullary rays are completely absent. Next, how can you remember this easily? The sequence of the description. Cats sit to play games. Cuticles, stomata, trichomes are absent. Protection and gaseous exchange for epidermis. Hypodermis, strong muscles because it has sclerenchyma in its composition and its function is mechanical strength. Then big, intelligent, very energetic. Big parenchyma cells are present in the ground tissue. Intercellular spaces are present. variable cell size, and endodermis is totally absent in the ground tissue. Vascular bundles here, how they are arranged, they are just scattered in the ground tissue, which is atactostele type of arrangement, and then sclerenchymatous bundle sheath, conjoined closed type of vascular bundles, tiny peripheral bundles, and phloem parenchyma is absent, xylem parts are present. And here if you see water filled protoxylem lacunae with no pericycle, no pith and no medullary rays. Clear, right? Mnemonics here are not that necessary, that is just for your understanding. Whoever feels comfortable in remembering the answer sequence with the mnemonic, you can go with the mnemonic. Whoever is comfortable with diagram and its explanation in a sequence, that is also fine. Okay, that is up to your comfort and your liberty beta. Next, if you observe, describe the internal structure of dorsiventral leaf. Dorsiventral leaf, and then we have, this is the last question of this chapter. Describe the internal structure of dorsiventral leaf with the help of NEET label diagram, asked multiple times like in March 2020 and March 2017. So, here if you see the TS of dorsiventral leaf shows three main regions. One is epidermis, second is mesophyll and vascular bundles. Here the cortex is mesophyll only, or the ground tissue system is mesophyll only. If you express the answer in the part of three tissue systems also, it is fine. Here under epidermal tissue system of dorsiventral leaf, only epidermis will come. Under ground tissue system, only mesophyll tissue will be included. Under vascular tissue system, vascular bundles are present. Okay. Here we go. Here you see, you have, first I will describe the complete diagram and then we will go for the theory. So, here you see my epidermis is covered by what? Cuticle layer, thick cuticle layer, which is composed of cutin. Upper epidermis is also called as, upper epidermis is also addressed as ventral epidermis, which is present on the ventral side, or adaxial epidermis. Upper epidermis, which is on the ventral surface, or this is also known as adaxial epidermis. Fine, right? Next, here you see below the upper epidermis, 2 to 3 rows of palisade parenchyma are present, 2 to 3 rows, which are vertically arranged with least intercellular spaces. Palisade parenchyma and spongy parenchyma, they both comes under ground tissue system. And on the lower surface, you have lower epidermis on the dorsal surface. This is also called as abaxial epidermis. If you look into the stomatal distribution, more amount of stomatal distribution is seen in the abaxial or dorsal epidermis, because for the prevention of transpiration. This is an adaptation to prevent transpiration. Okay, more amount of stomatal distribution is happening in the lower surface of leaf. And here if you see vascular bundles are surrounded by just single layer of bundle sheath cells, and vascular bundles consists of vascular bundles, that is xylem and phloem. They are of closed type and they are at the same radius and closed type of vascular bundles. Closed type of vascular bundles are present. Clear, right? Next you see. Epidermis. Epidermis is present on the both surfaces. Lower is abaxial, upper is adaxial side of the leaf. Epidermis, this is also known as lower epidermis and upper epidermis respectively. How cells are arranged? Cells are barrel shaped, compactly arranged, and epidermal cells, they do not have any intercellular spaces, they lack the intercellular spaces. Both adaxial and abaxial epidermis are covered by cuticle. The presence of multicellular hairs and minute pores is called as what? Stomata. Each stoma is surrounded by bean shaped guard cells or kidney shaped guard cells. They are found on both the surfaces but more numerous in the lower or abaxial epidermis and fewer you can see in the adaxial epidermis. And then we will see functions. What are the functions of the epidermis here? It provides protection to the internal tissues, regulates transpiration and facilitates the gaseous exchange. Mesophyll, as I told you, it is differentiated to what ma? Palisade parenchyma and spongy parenchyma. In dicot leaf, this arrangement gives the leaf its dorsiventral nature. Next, palisade parenchyma position, this is just located below the upper epidermis, below the adaxial surface. Cell shape is, it is having elongated cells with small intercellular spaces, and arrangement, they are vertically aligned parallel to each other. Chloroplast contains numerous chloroplasts. This is for photosynthesis. Spongy parenchyma, these are found towards lower epidermis or abaxial epidermis beta. Extension extends from palisade cells to lower epidermis. What is the cell shape over here? Oval to round shaped spaces, they will have large intercellular spaces for gaseous exchange. Next you see vascular bundles. Of course, vascular bundles are conjoined and closed, but as it is dorsiventral leaf, if you see, veins will vary in its thickness in the reticulate venation and the pattern of dicot leaves. Vascular bundles are large at the base of the lamina and smaller towards the margins of the apex, okay, large at the base and sharper at the apex. Bundles are conjoined, collateral and closed type. Xylem faces on the upper side, phloem will face towards the lower side. Vascular bundles are surrounded by a layer of thick walled cells called as bundle sheath cells. They are arranged compactly. Next you see ma bundle sheath extensions. They will grow towards upper and lower sides. They will help in conduction of food materials from mesophyll to vascular bundles. Okay. So, in dicots, bundle sheath cells are initially parenchymatous and they will later turn to what? Collenchyma. Main structure on mnemonic, if you see, SBCCSPF, where single layered on both sides epidermis, barrel shaped, covered by cuticle. This contains multicellular hair like projections. Stomata is with kidney shaped and P. They are present on both sides. Next, yeah. So, what you have to describe in a dicot stem and in a dorsiventral leaf. First, dorsiventral leaf, though upper and lower epidermis you describe ma, and then ground tissue system which includes palisade parenchyma and spongy parenchyma, and then you write about vascular bundles. So, in the anatomy of histology and anatomy of flowering plants, definitely there will be one question for sure. So, in every year, if I have to observe the previous year papers, there is question in most of the years from histology and anatomy of flowering plants. Highlight, please go through the dicot stem and monocot stem, dicot leaf, dicot root and monocot root. From these areas, you will get the question for sure. Okay. My advice is first you draw the diagram and then go on describing the diagram. Okay. Because once you draw the diagram, then the regions and the layers will be clear to you. So, you can easily describe that in a sequence. If you are still unable to present the answer in a sequential flow, then take the help of mnemonics which I have provided. Okay. Take the help of mnemonics and then present. And again I am telling you, the mnemonics which are provided in the book are just for your understanding and easy remembrance, but it is not to present for the exam. I am reminding you again and again. Okay. So, at last I would like to tell you one thing, that from morphology 3 questions we are done, from histology around 4 to 5 questions we are done. From these questions, long answer type of questions, definitely we can expect 2 when I have to analyze all your previous year papers. Okay. And after this we are going to start with the reproduction, sexual reproduction of flowering plants. From that also we have very long answer questions, after which we will move on to short answer type of questions and then very short SMA. So, this 60 days revision classes for IPE are completely for free. So to all the students, I advise is just go on looking into the video onto your comfortable tempo, okay, but then you will get an idea. Don't look at the question, its revision separately, IPE preparation separately or EAPCET preparation and NEET preparation separately, nothing like that. All the long answers, short answers and very short answers that you are preparing for, okay, they will be in a synchronization with your EAPCET and NEET as well, but that is objective type of answer, this is subjective description, that's it, everything else is same. So the way you are learning should be in combination, you keep in your mind about all the 3 exams and prepare the content, okay. So that will be helpful to you after your IPE exam when you go for EAPCET and NEET as well. So I'd like to wind up telling you that 2 chapters LAQs are done. You can go through the playlist, and in our next class we will be starting new LAQs, okay. Thank you.